Welcome to Biology Teaching and Learning video. This video will be explained about the effect of linked genes with crossing over on the dihybrid test-cross ratio. Before we begin discussing linked genes in detail, let's refresh our memory on what we have learned from Mendelian inheritance. According to Mendel, one characteristic is determined by a single gene. One gene has only two variations called alleles. Each gene located at different locus on different chromosomes, hence can assort independently. For example, the genotype of this individual is capital R small r and capital Y small y. Since the two genes, gene R and gene Y, are located on different chromosomes, the two genes can assort independently of each other's during meiosis, resulting in the producing of equal number of each gamete, capital Y, capital R, y, small r, capital Y, small r, and small y, capital R. So therefore, if test cross is carried out on this individual, the phenotypic ratio of the offspring will be 1 to 1 to 1 to 1. Now let's learn with linked genes. Linked genes refer to genes that are located close together on the same chromosome and tend to be inherited together. For example, gene G and gene N are located on the same chromosome, although on different locus. Since they are located on same chromosome, therefore they will always be inherited together and cannot sort independently during meiosis. Although we are looking at the inheritance of two characteristics at one time, since the two genes are located on the same chromosome, the way we write the genotype would be slightly different than how we would write in dihybrid cross. To write the genotype of linked genes, we use an oblique or slanting line to separate alleles on homologous chromosomes. For example, in this case, one chromosome containing capital G, capital N oblique, another chromosome containing small g, small n. Now, let's continue our lesson by discovering how linked genes can change the expected Mendelian ratios. If crossing over does not occur, it is very difficult to form recombinant gamete. So, it only produces two types of gamete, which are capital G, capital N, and small g, small n, unless the process of crossing over takes place, which is the exchange of genetic material between non-sister chromatids of homologous chromosome during prophase 1 of meiosis. To explain in detail, let's use these two genes as an example in a case where crossing over occurs. The genotype capital G, capital N oblique small g, small n will produce four types of gametes, capital G, capital N, and small g, small n as the parental gametes, and capital G, small n, and small g, capital N as the recombinant gametes. Even though these two genes are located on the same chromosome, they can be separated due to crossing over between non-sister chromatids of a homologous chromosome pair during prophase 1 of meiosis. This allows the formation of recombinant gametes. Let's say, in fruit fly Drosophila melanogaster, the allele capital G is dominant for grey body. Meanwhile, the allele small g is recessive for black body, the allele capital N is dominant for normal wing, and the allele small n is recessive for vestigial wing. The two genes located on the same chromosome but different locus. From this information, we quickly know that gene G and gene N are linked. This slide shows a genetic diagram of linked genes with crossing over. It illustrates how two genes located on the same chromosome may still be separated due to crossing over between non-sister chromatids during prophase 1 of meiosis. Although the genes are linked, crossing over can produce recombinant gametes, but only in small numbers. As shown in the diagram, parental type offspring are produced in greater numbers. Recombinant offspring are produced in smaller numbers. Because of this, the phenotypic ratio is not 1 to 1 to 1 to 1, unlike the ratio seen in a Mendelian dihybrid cross. This deviation indicates that this is linked genes. Let's recap what we have just learned at these examples. A test cross of on-fly with red eyes and normal wings give rise to offspring with the following phenotypes. We can see two big numbers and two small numbers. So we can be fairly sure that the two genes are linked genes. Another example, a test cross on plant with round yellow seed give rise to offspring with the following phenotypes. As you can see, the numbers are very close to each other, implying that the ratio following 1 to 1 to 1 to 1. So the two genes are unlinked, and this is dihybrid cross. That's all for now. Thank you for watching.